So to give you an idea of cost on this, so you'll see on here, it's got GIST Medical Advisory Board. We had a GIST Medical Advisory Board, so when we had research projects come to us to look at, we as patients and advocates aren't intelligent enough. This is why these guys have got names like doctor and professor and associate professor in front of their names. What we do is we had a collection of eminent people within GIST to look at the proposals, such as the ones that we received from the Southampton University. But we more recently have decided that ourselves and Sarcoma UK, we're doing the same things. So we've had meetings with them, and we've decided we're going to combine our strengths. And now Sarcoma UK, represented by Kathy, who's here today. Carly. So they then look on our behalf at these projects, and we've got limited resources. Sarcoma UK also have limited resources, but combined our resources to be able to go into projects like this, which is why this project is a combination between us, our financing or our funding and Sarcoma UK's funding, we can do more together. So it was a sensible move to make. The amount of funding that we most recently put together, and all our funding, the money, comes from donations. That's it. This project and the other 3, so 4 altogether that we did in the last round, our input to it was £145,000. Sarcoma UK's was more, because that's how we agreed to set the balances. So it shows you, I don't know about you, but this to me could be revolutionary for GIST cancer, and it wouldn't have happened without our funding, and Sarcoma UK's funding. So, you've invited us to go down to your labs and actually look at this happening in real time, and I'd love to do that. But I just wanted to emphasise how grateful we are as a community to you guys doing this stuff, and thank you for doing it. But as a charity, we are making sure that we're able to do this. So I'll end that there and ask if there's any questions.
Thank you.
Is there anything that we as patients can do to boost our immune system, or do we have to sit back and wait for your amazing research to produce the results and for the doctors to pump it into our bodies?
No, not at all. So we work on a lot of what we think is, these drugs, trying to boost NK activity. And actually there's some really interesting papers that come out a bit out of our field. It's things like healthy eating, Western diet characterized by high sort of fats that can all inhibit immune activation. And things like exercise. So there's just been a study published on natural killer cell activity, and they took some patients who had leukemia who just gave their natural killer cells, and they saw how well they could kill cancer cells in the lab. They then took another group of patients who had a round of exercise just before they took those natural killer cells, and the natural killer cells were able to kill much better after the exercise. So I think healthy eating and exercise is not only good for our bodies, it's also good for the natural killer cells.
Are puddings and ice cream and sweets, do they all help?
I tell my children not.
Oh, I'm talking of your children, but you just mentioned briefly your daughter, did you say?
I've got 2 sons, 6 and 9.
Okay, your 6-year-old son said that it was like a shotgun. He's the future, isn't he? That's the future that we're looking at. Any more questions?
I found myself thinking that was absolutely fascinating, and I wanted to ask that as a patient on tumour resection, can we ask to donate some of our cells to help your research? Would that be helpful to you? Is it possible? I mean, the more the merrier. If you're only getting your samples from just patients in Southampton, or can we request that,
so
and could it be helpful?
I would just say patient samples are really essential. So as part of this project, Dan has actually been collecting just patient blood samples with Jack and building up a stock of those, so we can look not only at healthy donor NK cell activation, but actually just patient NK, and that's really, really important. And as well, if we can look at activation against the primary tumours, that's really, really helpful. So we're really grateful for any donations. And I think Zoe is desperate to say something now as well, so I will just...
When you give blood or undergo surgery or whatever, you'll probably be asked to consent
and if you're asked to consent, then if you can sign that consent, then your samples will be taken. We work really closely with Robin and there's a National GIST Tissue Bank and when you consent, your samples will hopefully go into the National GIST Tissue Bank. So we're using samples from that GIST Tissue Bank for this research. So some of what Jack does, we're using tumour material from that. For some of what Matt talks about, we have to get it in a particular way, but again, we're working really hard with Eniola at the Tissue Bank at the Royal Marsden to make sure we get exactly what we need when we need it, but it's all about that consent, and I think Nick will be able to tell you a bit more about that.
So a number of years ago, Jane Bressington, who was vice chair here and runs the Paul's GIST Clinic, she wanted to set a tumour bank up. Now, we're dealing with human material. She was told, no chance. It's the worst thing you could say to Jane. We now, because of Jane's work, we now have a national tumour bank. As you said, we've handed it over to the Royal Marsden to deal with, and Eniola is the lead on that. Basically, that means if anybody has an operation and consents to a bit of their tumour being sent to the tumour bank, it's there not just for these guys, but for everybody that's doing research on GIST tumour, and they all know about it. A note of caution on it: we have plenty of exon 11 and all the more common exons. It's the really rare ones that we don't have enough of. So what I'd say, if anybody is due to have an operation fairly soon, contact us through our helpline or email, through our email, and we can put you in touch with Eniola in the tumour bank, and she will organise everything for you. We pay for it. We have to pay for it as, again, another part of where our fundraising goes, to ensure that tumour bank is kept topped up. Sounds really horrible, but you know what I mean. So, yeah. Any other questions?
Hello, thank you for your presentations. And I understand from last year and elsewhere that sometimes as a result of an inflammation caused by another illness, it can bump start the immune system into attacking a cancer. So if that's true, has that given you any inspiration in this work that you've done, or is there any link there?
Yeah, definitely. So it's known, I think that first picture I showed with all those different types of immune cells, and they don't just all work in isolation, like you say, they have to work together as a team. And the more of them you can activate at the same time, the better the response is. And that's really becoming more and more clear that we shouldn't be working on one cell type, it should be acting together. And I think, so something like imatinib, we've been looking at its effect on NK cells, but it can also affect other cells of the immune system, like dendritic cells or T cells. So we're looking at sort of integrating all of this to see how we can best boost that. So I think that's really, really important, is activating that broad response. And that's something that I think some of the drugs that are already approved might be already really good at, but we just have to time it really well to give some of them a bit of a head start and a leg up.
Hi, thank you for your talk. A bit of a technical question, but am I right in thinking PD-L1 inhibitors have been tried in GIST and they weren't particularly effective? How does that interact with your findings on imatinib?
Yes, so I think by targeting one immune checkpoint, I think like we were saying about activating multiple cells in the immune system, so I think that's PD-L1 is, the receptor for PD-L1 is expressed at really low levels on natural killer cells, but things like NKG2A are expressed on the natural killer cells and not so much on the T cells. So I think maybe inhibiting a broader range of these immune checkpoints simultaneously might be a better approach to be able to switch on more of the immune system at one time point. So I think we know from that, I think, that targeting one immune checkpoint probably isn't going to be effective, and you need this combination therapy. So yeah, that's really informed what we're doing and looking at this sort of multi-pronged approach. And I think we wouldn't ever be imagining we'd inhibit NKG2A alone. It would have to be with other things to try and sort of boost that.
Right, so thank you very much, both of you. I'm really excited by the research that's happening. I'm really pleased that we're able to help you by funding it. Thank you to everybody in the room, because it's, as I say, money from fundraising that pays for this. Keep up the good work, and let's organise a time that we can come down. And I don't mean everybody, by the way. We'll take some photos, we'll put it on the website, we'll do the stuff that Kathy and Ros do that tell you guys what all these things look like. So thank you, brilliant.